Good morning, everybody. Welcome to Pediatric Grand Rounds. Before we get started, just the usual reminders from me. Please stay muted during the presentation. If you have questions or comments for our speaker, please add them into the chat with everyone box at any time during the talk, and we will share those questions and comments with our speaker at the end of the presentation during the Q&A. You'll be getting an evaluation by email from our team sometime today or tomorrow. Please make sure to fill that out for us as it helps us with future content. And finally, just a disclosure from the planners of Grand Rounds, we have no relevant financial relationships to disclose. So for now, I will turn things over to Dr. Angela Sun from the Division of Genetic Medicine to introduce our guest speaker for today. Welcome, everybody.
Good morning, everyone, and thanks for this chance to share a really exciting topic with you guys. I'm Angela Sun from Biochemical Genetics, and our speaker today is one of the attendings that trained me and really helped me along in my career. So Dr. Raymond Wong from CHOC, Children's Hospital of Orange County, and he did his medical school education at UCLA, so a Southern California native like myself, and then went on to do a genetics residency or fellowship. I guess the term is now ambiguous, but in clinical genetics, as well as a fellowship in biochemical genetics. And he's currently the director of the multidisciplinary lysosomal storage disease program at CHOC and board certified in both clinical and biochemical genetics. So with that, I will let Dr. Wong take over.
Okay, thank you, Angela, for your introduction and for hosting me. It's an honor to be here, and honestly, I don't think of you as a trainee, but as a friend and a colleague. So it's great to be here. Here's my talk. And this is the first time I'm giving it, so I'm going to try and whiz through it so that we can finish on time. Here are my disclosures. Those are my, I'm going to do my very best to provide links to references and that kind of thing, although there's no industry influence at all here. Goals and objectives for today. So I'm going to use Pompe disease as an example for a rare disease that potentially has therapy with genomic medicine. So just to start off, I think you've seen some variant of this slide. Rare disease is defined as a condition where there's less than 200,000 people in the country that are affected. But when you put every single rare disease together, it ends up being that somewhere between 25 and 30 million Americans actually have some kind of rare disease. About 70% of them are inherited or genetic in etiology, and about 70% of them affect children or have onset in the pediatric age. And currently, really less than 10% of those have any form of treatment. And so it's kind of an area that is waiting for genomic medicine as a method of treatment. That means that there are about 12 million children estimated nationwide, about close to about 1 million in the state of Washington, and here in King County, about 225,000 children who have some kind of genetic condition that might be amenable to genetic correction. But as I mentioned before, I'm going to use Pompe disease as an example of a rare disease with potential for some kind of genomic medicine approach. As some background, Pompe disease was first described by Johannes Pompe, who was a Dutch pathologist reporting glycogen storage in a little girl who passed away from hypertrophic cardiomyopathy. It's a recessive condition, it's a lysosomal storage disorder, and a glycogen storage disorder. In the textbooks, it's estimated that the prevalence of Pompe disease is about 1 in 40,000 live births. The infantile onset, or the severe version, is a progressive and fatal condition that affects skeletal and cardiac muscle, as well as spinal cord neurons. The gene that is responsible for Pompe disease is GAA, which encodes an enzyme called acid alpha glucosidase, which I'll also refer to as GAA. And as you can see here, pop up my pointer, you can see that there are multiple, in addition to the striations of muscle, most of it is actually just full of like stored junk, which really interferes with proper function of the muscle. In terms of presentation, neonatal onset hypertrophic cardiomyopathy is the cardinal symptom for IOPD, or infantile onset Pompe disease, which presents as tachypnea, tachycardia, and poor feeding. And oftentimes, these symptoms go on for several weeks before the patient, the infant, becomes fully symptomatic. And so clinically, they would present to the office or the emergency department with florid heart failure. If you get a chest x-ray, you can see that the heart is massively enlarged. It is, in this case, more than 2/3 the size of the chest cavity, when really it should be 1/3 or less. When you get an EKG, you don't see many conditions that cause really, really tall QRS complexes, and that's from really the severity of the left ventricular hypertrophy. There can also be some pre-excitation, which you can't really see here. Skeletal myopathy is really the second cardinal symptom. And here's a patient of mine who you can see as he's walking up really demonstrates the classical Gowers maneuver of proximal myopathy. There's prenatal onset glycogen storage, and so this right here is a muscle biopsy from an infant in Taiwan who was diagnosed with Pompe disease by newborn screening. And so this was obtained at about 2 weeks of life. And instead of being nice and pink, you can see there are all sorts of white glycogen granules here. And then as time passes and the amount of glycogen really starts to build up, the amount of contractile elements in the muscle gradually declines until the muscle fiber is completely full of glycogen, becomes fibrosed and essentially non-functional. There's also some anterior horn neuron pathology, which contributes to the skeletal muscle dysfunction. Here's an old slide that describes the pre-treatment natural history. And so you can see that, you know, first signs and symptoms appear very, very early in life. Mean is about 2.5 months. But prior to newborn screening, diagnosis really wasn't until about 6 months. And you can see the children progress to respiratory failure and then death very, very quickly. So rapidly progressive skeletal and cardiac myopathy with early mortality. There is a form of a condition called late onset Pompe disease. These folks typically don't have any symptoms in infancy, but as the amount of glycogen storage progresses, there's some subtle but progressive proximal muscle weakness that really doesn't get noticeable until late adolescence or early adulthood. These young adults are often going to neuromuscular clinic in the adult world, and maybe misdiagnosed as having idiopathic limb girdle muscular dystrophy. Over time, as the myopathy progresses, there's respiratory insufficiency caused by weakness in the diaphragm and the accessory muscles. And so ultimately, what LOPD patients succumb to is hypoventilation and a combination of respiratory failure. This slide is also from Priya Kishnani, who's a glycogen storage disorder specialist at Duke. And you can see that first symptoms on average are in the mid-20s, although it can be very early. And then over time, proximal muscle weakness progresses until eventually patients are really requiring ambulation support and even BiPAP or invasive ventilation in their mid-40s, 50s. So regardless of early on infantile onset, late onset, Pompe disease is a progressive and serious sort of life-limiting condition. There is treatment for Pompe disease. Enzyme replacement therapy, which is essentially synthetic GAA enzyme, was approved by the FDA actually almost 16 years ago. It is given every 1 to 2 weeks. It has to be given intravenously. You can't take it by mouth. And the dosage here is anywhere from 20 to 40 milligrams per kilo. And in comparison to some of the other enzyme replacement therapies that we give for our lysosomal conditions, the dosage is anywhere from 20 to 40 times more than we give. So you have to address the muscle. It's a big tissue. A lot of it actually ends up going to the liver instead of the muscle. And so you have to give, a real whopping dose, which has some relevance later on. It is efficacious for reducing the hypertrophic cardiomyopathy symptoms. And so here's a young patient that presented to us at 2 months of age with cardiomegaly, with heart failure. And we started her on enzyme replacement therapy. And here she is about 3 years later. You can see the portacath that we've had to install because of the recurrent infusions. Her heart size is definitely reduced and normalized, but she still has residual symptoms of skeletal myopathy. When we did an echocardiogram to measure the left ventricular mass index, you can see that normally it's about 90 grams per square meter. And so here's where she was sort of pre-symptomatically. And then before treatment, her LVMI had actually increased to almost 4 times normal. And we started enzyme replacement therapy, and rapidly it came down, not exactly to normal. It took about another 1.5 years to normalize, but we were able to get her out of the woods, out of the cardiac ICU, where she was in pretty significant heart failure. It does slow, but it does not prevent the progression of skeletal myopathy. So that young lady requires AFOs, has some proximal muscle weakness, and she also has some upper airway oropharyngeal muscle weakness. So she has like characteristic velopalatal insufficiency and kind of myopathic facies. When the initial clinical trials were done, this enzyme replacement therapy was shown to increase ventilator-free survival for both infantile and the late onset versions. But there are pretty significant limitations. This is a condition that has to be treated lifelong. Enzyme replacement therapy is not permanent. It's like you have to fill up your gas tank every few weeks, right? If you run low. And after a while, you really need central line placement, and there are complications associated with that. The degree of dosage that we're giving exposes the body and the immune system to a significant amount of antigen. And so it's not response, it's response. The humoral immune system actually reacts to the enzyme, creates antibodies, and in certain cases, the amount of antibody that's generated is enough to even impair the efficacy. And so we oftentimes now actually have to treat our infants with rituximab and methotrexate and some pretty heavy duty immunosuppressives to prevent this immunologic response. And as I mentioned before, most of the infused enzyme ends up going to the liver and not the muscle. And so really, you're not treating the organ that's affected. It's getting shunted off somewhere. So we'll talk about other approaches to treatment in a little bit. I do want to talk about newborn screening, which is a fairly new development in the state of California. It wasn't implemented until about 4 years ago. And I think Washington State's been doing Pompe disease screening for a little bit longer. Really, it piggybacks on the tandem mass spectrometry technology that's used for newborn screening for other metabolic diseases. Essentially, you start off with a blood spot, a dry blood spot, a little punch is taken out of it, and it's run with an artificial substrate where the more enzyme activity there is, the more analyte that's released, and then you can measure this with tandem mass spectrometry. If you find a low analyte level, that actually corresponds to a low enzymatic level, and so then it can get flagged and ideally lead to an early diagnosis and onset of therapy. In reality, it's a little bit messier, so you can see that most folks are within normal range, but then there's this overlap area where some normal patients or carriers may live here, and that overlaps with the late onset patients or the infantile onset patients. And so, as you've probably experienced, the reality of newborn screening is a little bit messier than the ideal. Just for a little bit of experience from other states, over a 5-year period, Missouri screened almost 500,000 newborns. There were 0.05% of newborns were recalled, but out of those 274, only 46 were actually confirmed to have Pompe disease with a 17% positive predictive value. Of those 46, actually only 10 had late onset Pompe disease. So that actually kind of fits the textbook prevalence about 1 in 40,000. However, 80% of these cases actually ended up having late onset Pompe disease. So then we're stuck in a situation where, we have infants that aren't going to be symptomatic, may develop symptoms in anywhere from 10 to 40 years, but now you have very, very anxious, worried parents. And so what do you do from there? To top that off, in the genomic era, there are always going to be people who have genetic sequences that have never been seen before. And so what do you tell those families where you say, well, we've never seen your child's mutation before. We don't know if it's going to be significant or not. And so we just have to follow your child over time. That creates what we call a patient in waiting kind of category, which can be a little bit nerve-wracking, actually a lot bit nerve-wracking for our families. In addition to that, this technology can actually pick out carriers, what are called pseudodeficiencies, where there's a change in the gene that causes a reduced enzymatic activity in the test tube, but not in the patient themselves. And the rest were just false positives. So, like I said, a bit more messy than the ideal. In California, also almost 1 million newborns screened over the period of a year, 0.02% recalled. only 18 confirmed for a 20% positive predictive value. 2 had infantile onset Pompe disease, 11 late onset. So again, that 5-to-1 late to early onset ratio. And again, 5 genotypes of unknown significance, a whole bunch of carriers and pseudodeficiencies identified, and a few normal. So what do we do if we get a newborn screening case in California? Thankfully, a lot of the work is actually done for us on the state level before it comes to the metabolic doctor's office. So an infant is flagged if their activity is less than 18% of the daily median. From there, the state actually automatically extracts DNA from the blood spot and does genetic sequencing. Thankfully, if it's a carrier or a pseudodeficiency, we are not contacted, and they're just automatically flagged as false positives. However, if there are 2 or more genetic variants in the GAA gene, whether they're pathogenic or uncertain, then it gets passed on to the pediatrician and the metabolic doctor's office. What we do when we get one of these calls really kind of depends on the enzymatic activity. If it's super, super low and potentially suspicious for infantile onset Pompe disease, we'll actually call the family and ask them to present to the emergency department for these laboratories and for an echocardiogram, EKG, clinical evaluation, and a chest x-ray. And, you know, if there's no cardiomyopathy, if the baby looks good and is feeding well and not hypotonic, then, you know, we follow the confirmatory labs very closely. But if there's hypertrophic cardiomyopathy, these children are often just directly hospitalized. If the enzyme activity is low, but not low enough to trigger like significant alarm, we'll have the family present to either the hospital or a local laboratory to get a CK to look for myopathy, transaminases, which are often elevated in conjunction with CK. And then there's a biomarker called hexose tetrasaccharide, or HEX4, which is a biomarker of storage. And so if there's active glycogen storage, this will be elevated. We typically don't see HEX4 elevated in the late onset babies. It's only in the infantile onset babies that have an elevated hexose tetrasaccharide at birth. And so that's another kind of discriminant between infantile and late onset Pompe disease. What we do next, and so if the diagnosis is more consistent with late onset, what we'll do, actually for either case, there's a significant amount of reassurance that goes to our families. As you can imagine, whether you tell them not to Google things or not, the families are going to look it up. So we actually try to direct them to the right resources. But before that, if you look up Pompe disease, it's really kind of scary. And so a lot of what we do is reassurance, especially in the case of LOPD, we distinguish the 2 conditions, and then we say, look, you know, we're able to prospectively have their children followed by a physical therapist, typically do a gross motor assessment starting at about 6 months of age, and then reassess as frequently as the PT thinks is appropriate. I don't typically start enzyme replacement therapy in the late onset Pompe babies because taking a small child and having to give enzyme replacement therapy, you know, every week is just too much for a condition that may have an onset at age 20 or even later. The evidence does indicate, however, that a high protein diet does prevent or slow down the progression of myopathy. The thought is potentially you're providing the muscle with lots of amino acid substrate to prevent atrophy. And so we have our metabolic dietitians involved for augmentation of protein in the diet. And then we see our babies typically every 6 months, although if there are parental concerns, we do ask them to contact us and we'll evaluate them earlier. The initiation of enzyme, well, we've only been doing this for 4 years, so none of our LOPD babies are currently on enzyme replacement therapy, but typically if there are concerning signs of myopathy, then we would consider starting. For an IOPD baby, we will start multidisciplinary follow-up, including pediatric cardiology, often orthopedics, because the hypotonia does induce ankle, contractures or scoliosis or other things. Physical therapy for sure. Metabolic, we often involve ophthalmologists because there's ptosis from the hypotonia of the eyelid muscle. We initiate enzyme replacement therapy as soon as possible, and oftentimes in the context of being in the intensive care unit. That's how sick these babies are. And weekly follow-up and hospitalize if there's heart or respiratory failure. Okay, so from the clinical aspect, Pompe disease is caused by a deficiency of acid alpha-glucosidase deficiency. It results in glycogen storage in the muscle, which then interferes with muscle function and contractility. There is an attenuated late onset form. Although storage does begin in infancy, symptoms often don't begin until adolescence or adulthood, and the LOPD folks don't have cardiomyopathy. There's the classical infantile onset form, which is a severe, rapidly progressive, and fatal skeletal cardiomyopathy. Although enzyme replacement therapy was approved in 2006, there are significant limitations in the efficacy. It really does address the hypertrophic cardiomyopathy if started early enough, but it really only slows the progression of the skeletal myopathy. And finally, given significant morbidity and mortality of infantile onset Pompe disease, the existence of newborn screen technology to measure GAA enzyme and the existence of available therapy, this condition was added to the recommended universal screening panel in 2018. And as you've seen, there are now significantly more late onset Pompe disease pre-symptomatic babies identified than the infantile onset kids. Okay, so the rest of my time, and I know I have limited time, so I really need to breeze through this. We'll talk about genomic medicine approaches. And you may have heard about CRISPR, gene editing. It's really hit anywhere from Nature now to the lay press. And so I wanted to give sort of a 30,000-foot overview of what CRISPR is, because we work on that in my laboratory. So what is CRISPR? It's an acronym for Clustered Regularly Interspaced Short Palindromic Repeats, which is why we call it CRISPR. It was actually inadvertently discovered in bacteria, and in retrospect, it's a way for bacteria to respond and essentially eliminate viral infective threats. And so these enzymes are actually nucleases. They cut DNA. But Jennifer Doudna and David Liu, who are at Berkeley and Harvard respectively, found that they could actually harness that to potentially serve as a find and replace, if you will, by providing these nucleases with the right search string, if you will. You could find the right place in the genome, make a few cuts, and the excitement really came that you can then potentially correct areas within the genome. So just to really, really quickly introduce how this enzyme works. Most of our CRISPR technologies use what's called Cas9. There are actually like 14 or 15 different Cas enzymes. But Cas9 is really nice because it's only one protein. The others are sort of multimeric sort of complexes, and Cas9 is easier because it's just one protein. In order for it to be equipped with this find and replace function, you have to provide what's called a cRNA. And essentially what it is, or a guide RNA, is a 20 nucleotide sequence, which is complementary to the DNA that you're searching for. And so essentially you give your guide RNA here, and then you plug that into the Cas, and it will essentially go around the genome and search for the corresponding 20 nucleotides that match this, which makes it really powerful because, you know, there are 4 to the 20th possibilities. And so it makes it very, very specific if you provide 20 nucleotides. Once it finds that area, it will actually make a nice little cut. And from there, you can harness various different mechanisms to fix potential mutations that cause disease. So with that, you know, CRISPR-based genome editing really is the ultimate in personalized medicine. So as mentioned before, the Cas9 enzyme will cut the DNA at a specific site. You target it to seek out like disease-causing mutations. And then from there, once the cuts are made, you can have different approaches to repair. And so what I'll do in our remaining time is really kind of introduce these to you. First is what's called homology-directed repair. And so if you provide like a template sequence, the body can then use that template sequence to edit in the desired sequence. And then there are 2 other sort of exogenous engineered tools. One is called base editing. The other is called prime editing. And really the end result is to create a corrected nucleotide sequence. There are certain advantages over enzyme replacement therapy. The first is, you know, the holy grail is you want to give a dose only once, and that permanently cures your patient. And so because you're fundamentally correcting the DNA sequence, that really holds the promise for permanent correction of the disease. Second, making it specific to whatever tissue that you're aiming for, whether it's the heart or the muscle or the brain, really avoids issues with delivery to unaffected or, you know, asymptomatic tissues like the liver. And then because all of this is happening inside the cell and it's not in the circulation, it helps to avoid issues with immunologic rejection and antibodies and all sorts of things. So let's start with HDR. As I mentioned before, you know, you have to provide a guide RNA, which essentially tells the Cas9 enzyme where to go. And then from there, it will cut the sequence right here and right here. And then what you want to do is to provide a donor template, right? And then the body's own DNA repair mechanisms will take this donor repair and then plug in what you want. Unfortunately, what we've found in practice is it's not quite that simple. There's this other repair mechanism, which is called non-homologous end joining, which basically uses your other DNA repair mechanisms will ignore the template and just randomly just stick those 2 broken ends together. And unfortunately, that process is kind of messy. So it will cause deletions or insertions, which really aren't that good for your gene because you're messing with the the way that the gene is read. And so back in 2016, when we started our CRISPR journey, this was the only tool we had, and we found it frustratingly difficult to get the cell to go with the HDR method. All we found were mostly indels or insertions or deletions. So because we have a patient with this particular mutation, 1826 Dupe, and so it actually inserts an additional letter A and totally disrupts the reading of the gene and the enzyme function, we thought, okay, let's try this HDR. And so this is work that was done by Jeffrey Huang and Chloe Christensen. We actually have mouse myoblasts that have this 1826 Dupe mutation. You essentially plug in the HDR components here and nucleoffect them into the cell, give it some time to work, and then sequence. Not only that, we can also look for enzyme activity because this mutation actually causes the cells to make zero enzyme. And so if you see enzyme, that's very encouraging that the repair is working. And so unedited, you see these two letter A's. Ideally, what you're looking for then is just one letter A, as it should be, and the presence of enzyme. Unfortunately, what we found was very, very low amounts of editing, and so we didn't even get to the point of measuring enzyme activity. So what we did see was, and sorry for the resolution of this graph, I always tell people to pay attention to the y-axis because you're like, oh, this is a really tall bar, but the most we ever got was about 7%. And really, the worst we got was 2%. And the problem with this not only was that there was very low correction efficiency, there were abundant undesired insertion deletion mutations. And so let me just orient you here. The extra A that's mutated that we want to correct is right here. And so really what you want to see is like, going from 2 to 1. And instead what you see is a whole mess of like different colored bands here. And that's because the Cas9 gene is, the Cas9 protein has just caused all sorts of havoc. And so, first do no harm, right? We ended up abandoning this approach because we knew that it would cause more problems than helping. So we abandoned that, we move on. Actually, I'm going to reintroduce this with the prime editing, but I do want to talk about something called base editing, which actually was developed, I want to say about 3 years ago. So the field of CRISPR has just really advanced very, very quickly. Instead of using the Cas9 enzyme to cut DNA, really what it does is that it uses the guide RNA to seek the proper DNA sequence. And so that's your find. And instead of making cuts, the people at Harvard basically tacked on what's called a deaminase. And what that does is that it will take a C and turn it into a T, or vice versa. It can take a T and turn it into a C. There's also adenine deaminases, which convert A's to G's and vice versa. And so the strength of this is that you can make very, very precise edits without necessarily cutting the DNA. The only weakness is that if you have a mutation that, for example, turns an A into a T or vice versa, you can't convert that. So we have a patient who has this particular mutation. You can see it's a C to T, and so the base editing aspect will really work. So we selected this as a candidate. And this is a bad GAA mutation because it creates a stop codon, essentially says, stop making protein. And so that truncated GAA never makes it to the lysosome. It has no enzymatic activity whatsoever. So we actually were able to get skin biopsy specimens from our patient. We add the base editor as well as the guide, which is the search function. We put it into the cell and give it some time to do its work, and then do the sequencing as well as the enzymatic activity. And what we're hoping to see is a conversion of this unwanted C into the unwanted T into the C that we actually want. And so you can see here, this is our patient who's a heterozygote. And, C is red and T is red and C is blue. We don't want red and we want blue, okay? This is the untransfected version. After 4 days of base editing, we're starting to see less T, which is encouraging, and more C. So we're like, yay, this is really exciting. And then after about a month, we actually saw no more T and all C. And so that was really, really exciting. So with this, because this patient is a heterozygote, the unedited form has about 50-50. And over time, at 4 days, we saw actually about 80% editing. And at about 35 days, it actually became 100%. And so you can see that actually even at about a week, we saw complete elimination of the mutant T and a correction to the proper C. And correspondingly, we saw an increase in enzymatic activity, which is really, really exciting because at baseline, there is 0 enzymatic activity, and we're actually able to bring it into the normal range. So the next steps for that is to do something called off-target analysis, and that's to make sure that we didn't accidentally edit somewhere else in the genome that's not wanted. We want to be able to package the base editing into a method that can be delivered intravenously. We want to do mouse model preclinical trials, so we're actually starting with that very, very soon. And then hopefully, the idea is to go from patient to bench and then back to the patient. So to be able to help our patients. All right, we're almost done, I promise. Last is prime editing. And so this is actually a fairly recent development from 2019. And the idea here was to be able to correct those mutations, insertions, or deletions that base editing couldn't address. And so to make a long story short, essentially what prime editing does is that it provides, again, the search function with this guide RNA, but then you add an enzyme here called RT, or reverse transcriptase. And essentially what you can do is that you can provide a template which tells reverse transcriptase what kind of sequence you actually want. And so you can basically, you see here's your mutation here in red, you provide the proper sequence here in green, and then reverse transcriptase will then edit in the mutation that you want. So you go from your mutation here in red to your corrected sequence here in green. This is a really busy slide, and essentially the idea of this was doing prime editing for various different mutations caused a lot of correct edits, which is in blue, whereas using HDR really caused a small amount of correct edits and a whole bunch of insertions and deletions, which are unwanted. And so what these authors basically posed was that prime editing was a much more accurate method of genome editing. And so because our HDR on this 1826 Dupe was horribly poor, we turned our attention to prime editing as an alternative. And really, it's the only way that we can address duplications, deletions, and these transversions that base editing can address. So the setup is very similar. This was done by Alessandra and Khan, who measured all the enzymes. Essentially, we would take the prime editing components, transfect them again into our mouse myoblasts, and we actually now have a human fibroblast line. Give it a few days and then look for ideally that correction and synthesis of enzyme. Thankfully, we've gone through several cycles of this. So using our first guide RNA, we were able to get about 12% efficiency in a small amount of enzyme. And so Alessandra tweaked things a little bit and was able to get our editing efficiency up quite a bit to about 50% and about 13% enzymatic activity. And then eventually we're able to get it up to about 75%. So it takes a few tries. It's not as simple as the lay press seemed to indicate, where you kind of like mix everything in and voila, you get what you want. There is quite a bit of iteration and tweaking that's involved. So satisfyingly, you can see this duplication here. This is the unedited version. And then the first one, okay, you know, there's a little bit of removal of this green A here, but you can see sort of all this other mess here. And so that's why Alessandra iterated, and then you can see more and more of the wanted C and the unwanted A. And then over here, it's just a function of your desired sequence. And so we actually started seeing more of what we wanted and less of what we didn't want. And this one right here is actually an intended edit. So that was really satisfying, and I'm sorry Con and Alessandra are in the way, but we're actually get up to about 25% enzyme, where before there was 0. Over time, it's sustained, and so you can see that with this pegRNA3, it starts off at about 50%, and then it might decline a little bit, but it really kind of levels off after 20 days, and the amount of enzymatic activity that we can get is pretty sustained. So, you know, to have late onset Pompe disease, you have about 2.5%. Getting you up to about 15% really should be able to get rid of all the symptoms. And this is just proof that we are able to get the prime editing components into the cells because they glow green. Next steps, again, very similar. We want to do off-target analysis. We want to be able to package them into a form that we can deliver intravenously. We were able to get an NIH grant to do the preclinical studies for the mouse model. And so we're actually starting to breed the mice right now, with, again, the hope of going to human clinical trials soon. So, promise, just a few more slides. There's some issues with Basin prime editing, right? So each mutation ends up becoming its own therapeutic product. And although this is really personalized medicine, literally at its best, the problem is every single one that you create for every single mutation, every single gene, ends up having to go through the whole regulatory process by the FDA. And really, you know, having to do a single, an application for every single, you know, guide RNA and every single method would really get pretty onerous. And so what we're really aiming for is a one-size-fits-all approach to genome editing. And this actually literally just came out a few months ago, is something called twin prime editing. And so I'm just going to, again, give a very 30,000-foot view of this. We haven't done this in our cells yet, but we're excited to do this. There's actually another sort of DNA enzyme called recombinase. And essentially what that does, if you provide a proper sequence, a recombinase can then put that in. Problem is, a recombinase needs to have a couple of what are called recognition sites. And so again, what the folks at Harvard did was to use prime editing to put in these recombinase recognition sites. And so if you put a recombinase site here, and then you add your DNA sequence that you want, plus the recombinase, then it will essentially stick this proper DNA sequence in front of your recombinase site and clip this out. And so it doesn't matter where your mutation is, whether it's an deletion or an insertion or whatever, you can actually take the entire gene and stick that in front of the site that you want. And so this is pretty exciting, but again, we'll see if it's as good as advertised very, very soon. So, conclusions. Introduction to genome editing. Number one, it requires something called a Cas nuclease. CRISPR requires what's called a specific guide RNA or an sgRNA to seek the proper sequence within the genome. Back in 2016, only homology-directed repair was available, which resulted really in low efficiency and very, very high proportion of unwanted insertions and deletions. And current viable editing methods include base editing, 2017, prime editing, developed in 2020. And so our lab was able to demonstrate proof of concept for sustained and on-target CRISPR editing using base editing with 100% correction and prime editing with 50 to 70% correction. And so really our hope is that, in the next few years, we'll have some clinical trials for Pompe disease. So just wanted to thank the folks in my lab and our animal healthcare technicians. Here's our funding sources, and I'm happy to take questions. Thank you.
Thank you so much. For everyone who has questions, you can feel free to either unmute yourself and turn your camera on and ask your question live on WebEx, or type it into the chat box, and I will keep an eye on that and ask your question for you.
Speechless. Now you can all become genome editors yourself.
From Russell Van Dyke, how does the IV infusion target the muscle/heart?
Yeah, that's a good question. And so there are currently a couple of competing methods for delivery. The first is with adeno-associated viruses with specific tropism for muscle. Problem with AAVs is that you can only package about 3 to 4 kilobases of DNA information, and these CRISPR components are much bigger than that. So we're looking also at lipid nanoparticles, where you can package the entire protein in without having to worry about space or length.
Thank you. And from Ellie Nisbet, amazing science. Thank you for this presentation. Agreed.
Thank you.
We have a few more minutes, so I'll just be quiet and wait to see if there are other questions.
Yeah, I have a
Somebody's
quick
raising their
question.
hand. Yeah.
It's me. So
Oh, hey, Dr. Lee.
this is a fascinating discussion, and so thank you for the improved education and understanding of CRISPR. It feels like this is an area that has a lot of ethical considerations. I mean, are there do you have guidelines for like when can you ethically use CRISPR, and like how good, obviously if you're very accurate then it's good, but then it's like, I think that it's one thing to say I'm going to repair a gene that will cause severe harm, but are people also thinking about, oh, okay, I'm going to try to make better people?
Totally. And certainly genome editing of any kind raises all sorts of ELSI questions. There was that doctor in China that got himself in hot water for, genome editing embryos. So I think, there's pretty universal agreement and really just to edit sort of pathogenic or disease-causing mutations. But you're right, I think there are some early forays into creating sort of an ethical sort of framework for, not doing, taller person, smarter, faster, all sorts of things. There are some early forays into that, and definitely, we don't want to be creating super people and that kind of thing with genome editing for sure.
Thanks for the presentation.
Ronnie, hi.
Good to see you.
Good to see you.
Yeah. Ray and I were residency classmates, so it's great to see you on the screen. Thanks for that presentation. I'm curious about, can you talk a little bit about, in terms of newborn screening, particularly when there's, in some sense, there's a lot of families with, such a low positive predictive value that are having a lot of undue stress to it. What's the thinking about, like, what is the cutoff? When is it not no longer helpful, but more harmful?
Yeah, I think everybody entered into this with the best of intentions. But I think whenever you have a disorder with very low prevalence, in order to catch all the true positives, you are going to end up inevitably generating false positives. And I think that's where you set your cutoffs, right? And if you want, I mean, this really does become, and I remember you're an MPH. So, if you set your cutoff to catch everybody that's affected, you're going to create a lot of false positives. But if you set your cutoff low enough that you don't have false positives, you may miss a few people. And so that really becomes, a really sticky place where, well, do you really want to diagnose everybody? Or do you want to leave a few so that you don't create so much stress? And believe me, we've had to talk so many families down from the roof because you look on the internet, I would do it. And I think truthfully, the best way to address this is to, really, really carefully select the conditions that you screen for. I think certainly there's technology to screen all sorts of conditions. I mean, so SMA and cystic fibrosis and all sorts of conditions. But I think lysosomal storage conditions were a bit controversial because the prevalence was so darn low that inevitably you're going to create a lot of anxious families. And a lot of my time is spent talking them down. So I don't know if that's a solution to your question. It's more like, well, here we are. This was, something that the legislatures decided, and this is all we can do. So really, it has to be decided on the legislature side, not on the medical side. Sorry. Something popped up and then it disappeared.
Yeah, we have time, I think, for this one last question. Arthur, I don't know what your last name is, but this is from someone named Arthur. Thank you for your talk. Slight variation on Russell's question. Can you comment on potential delivery mechanisms for the nervous system, peripheral versus central? I recall a few years ago, there was conversation around HSV, but I wasn't sure how this has been changing over time.
Yeah, I think so. People are always developing better, more specific and less, what we call off-target methods of delivery. Right now, the flavor of the month seems to be adeno-associated viruses. I think HSV is proven to be a bit more problematic because it's a bit more difficult to package and to eliminate sort of the pathogenic viral particles. I do know that there are AAVs that specifically target, say, the central nervous system. They'll cross the blood-brain barrier. They have tropism for the peripheral nervous system. But again, right now, the limitation with AAVs is the size. And I think people are working on various ways to cross the blood-brain barrier or just mechanically bypass the blood-brain barrier by, doing lumbar puncture, cisterna magna, or intraventricular deliveries. Good questions.
Really good questions and perfect timing. Thank you so much, Dr. Wang, for being here, for visiting Seattle, and for all your expertise in your work in this area. So much food for thought for everyone today, and we really, really appreciate you. And thanks to our audience for sticking with us, and we will see everybody next week. Have a great day. Have a great
So
Thursday,
much.
everybody. Thank you so much.